So today we're going to have a very detailed look at the microscopic functions and structures of the muscles, and then on Wednesday we're going to take a more overview of some important muscles, and then we move on. Now, as I promised, okay, which of the following correctly lists the 3 structural types of joints? So we know the answer for that. What is the answer? So everybody got C? Yes. All right, so there's fibrous, cartilaginous, and synovial. Now, I'll figure out sometime in the near future why this clicker is not working for this class. So here we go for the muscle chapter. So this is chapter 9. We're going to have first an overview on the characteristics of the muscles, different types of muscles that we have in the body. And we had briefly seen this before, right? So what we're going to focus on here is really the function on how it is at the macroscopical level that the skeletal muscle is working, okay? So first thing, we're going to take a brief look at the gross anatomy of the muscle. Then we're really going to focus on the microscopic aspects of the muscle fiber. We're going to look how is it that a nerve impulse coordinates the movement at a muscle fiber. And then how is it that a fiber is contracting? And basically, if we get to that part by the end of this session, I'm happy. All right, so here we have a skeletal muscle. All of our skeletal muscles are under voluntary control. That's your somatic nervous system, okay? So it's voluntary control. So here, skeletal muscles are considered to be organs. And why is that? More than one type of tissue. Yes, it has more than one type of tissue in there. So they are organs. Now, when the muscle moves something, you have to have this something to have the origin at some place. So the attachment of the muscle is to something that is not moving. And then the thing that moves is really related to the insertion point of that muscle. So if it doesn't move, that's the origin. It's like we see the humerus being the origin, and then the brachii muscles are going to move the lower arm. Okay. So looking at the whole shape of skeletal muscle fibers, we see they have a lot of stripes, and those are formally called striations. So we have seen that before. Now, when you have the contraction of the muscle, we know that is going to happen very quickly, and a lot of times it's going to be a very powerful and synchronized type of contraction. Now, that was for the skeletal muscle, right? So it's striated. Another thing that's very important for skeletal muscle, as we're going to see in a lot of more slides, is that one muscle cell, one skeletal muscle cell, which is also called a fiber, has a lot of nuclei and is very, very, very long. That's why it's called a fiber. Okay. Now, if you compare that to other type of muscle, like cardiac muscle, so we're going to see that it also looks striated, but it doesn't have a lot of nuclei. It has one nucleus per cell. And also, it's not a very horizontal form of striation. When you look at cardiac muscle tissue, you see that the cells have this conformation that they branch, they tend to branch instead of being one parallel organization of these fibers. So we see that cardiac muscle is only found in the heart. Of course, that's why it's called cardiac. Of course, it's also striated. And another very, very big difference between the skeletal muscle and the cardiac muscle is that the cardiac muscle works by involuntary action. Thank goodness. If you have to think every time you have to contract your heart, I think we'll be dead by now, right? Imagine contract, 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 contract. You can do nothing else but to coordinate the contraction of your heart muscles. That's not very possible, right? So it is really involuntary control. Now it's really interesting because the heart itself has its own pacemaker that coordinates the contractions of all these cells at once, all these fibers at once. Okay, so the other type of muscle fiber is a smooth muscle. This is also under involuntary control, and that really is found in their internal organs. Mainly is found in the walls of these hollow organs like the stomach and urinary bladder and airways and basically things that are supposed to be happening on their own without us having to think too much about them or at all. Also, we find them in large arteries. The arteries have also a smooth muscle. So here's a little summary putting all the three types of muscle fibers in one slide. So know the difference. And here would be another question for you, but everybody in the class is going to get the freebie points, right? So let's just answer this question. One of the following muscle types, which is the only one subject to conscious control. What is the type of muscle? Is the B, skeletal muscle. Okay. All right. Now, muscles here, we're listing 4 important functions of the muscle. Of course, number 1 is movement, very, very obvious, right? So number 2, function of the muscle is also maintaining posture, okay? And a lot of times we don't think about that as a voluntary type of muscle control, but if you think a little bit more about it, like if you're sitting up, you are really contracting your muscles to maintain yourself sitting up. Have you seen what happened to somebody who stops maintaining control of their toning up posture muscles? Like, for example, by falling asleep, what happens to them? They kind of slump down and don't stay sitting up, usually, right? Although I've seen people in New York sleeping while they're standing up on the subway. Don't ask me how that happens. I don't really know. But usually you have to be awake and conscient to be able to keep your muscles contracting. And another function for the muscle is really maintaining posture. So a very important function is also stabilizing joints, right? So the joints will not come out of place. And another important function of the muscle is generation of heat. And that's a bit obvious too, because well, not that much in South Florida, but when you go out in a very cold temperature, what's the first thing that starts to happen to you? You start to shiver, right? So you're outside and you're like, I'm shivering. Now, question, can you stop shivering just by conscious control? Yeah. All right, go for the vote. How many people think yes? How many people think no? So people who think no actually never have been, I think, in very cold weathers, have you? Maybe? You can stop shivering by conscious control because it's under your voluntary muscle function. Those are striated muscle tissue, voluntary muscle tissue. Okay. Okay, so characteristics of muscle tissue. So basically, the muscle has to be responsive to something. So that characteristic is called excitability. Okay, so the muscle has to receive some kind of stimulus and it should be able to respond to that stimulus. And of course, contractility. Once you send a signal to the muscle, it is supposed to contract at will. Okay. Extensibility. What is this word? What does this word mean? The muscle can be stretched, but not too much. It's a little bit of a stretch, right? You cannot extend the muscles too much. And then elasticity, which also goes together with extensibility, is that once you contract the muscle, you want the muscle to recoil to its original position, original size. So that's another very important characteristic of muscle. Because imagine if you just contract the muscle and then it just stays there, right? So it's like kind of counterproductive. Okay. And actually, I do have a little story about that. Coming back to functions of your muscles and what is it that you have to know. So basic muscle anatomy. First thing, when you look at the muscles, you're going to see that it has a lot of nerves. It's very innervated, and you have a lot of blood supply. Why? Why do you need blood supply for your muscles? It needs a lot of oxygen, right? You have to have a lot of power, a generation of power in your muscles. You have to have a lot of ATP. So you need to have a lot of oxygen for your oxidative use of energy. So here we have a lot of blood supply, a lot of nerve, and you also have this connective tissue, sheets and attachments, because of course without the connective tissue, then your muscles would not be connected to the bones and they would basically be useless, right? Now, looking here at nerve and blood supply, so you know that each muscle is going to receive a nerve, artery, and veins, because of course the artery is going to bring the blood, the veins is going to take out the deoxygenated blood. Okay. So looking at the connective tissue sheets that are in the muscle, we see from outside to inside, you have a sheet that is called the epimysium that is on the very much outside, like a whole glove kind of surrounding the muscle. So this type of connective tissue is dense, irregular connective tissue. Okay, so dense, irregular, because it really has to distribute forces throughout this tissue. So this is going to surround the entire muscle. Then you have the perimysium, and this is going to be more to the inside of the muscle. Of course, we do have pictures to show that. Okay, so then you have this perimysium. They're going to be surrounding the fascicles. And as we see, fascicles are groups of muscle fibers. And then you have this endomysium. And this endomysium is different than the other two connective tissues. So epimysium and perimysium, those are both dense, fibrous, irregular connective tissues. Now, endomysium is fine areolar connective tissue. So this is different than the other two types of tissue. And this is going to surround each muscle fiber, okay, individually. So here's a big picture of where you find this connective tissue. You see on the outside, like surrounding the whole muscle, you're going to have your epimysium. And then when you look here into this bundle of fibers, what's the name of the bundle of fibers? Fascicle, yes. So the name of the tissue that is surrounding the fascicle is called endomysium. And then each muscle fiber, you're going to have, okay, perimysium, endomysium, and I lost it. Epimysium, perimysium, endomysium. There we go. From outer to inner. So endomysium is in the inner the fiber, and then you go out. Is this here is a perimysium, and then you go all the way out, and this one's going to be your epimysium, the outside. Okay. Do you have to know those connective tissues? Yeah, those are high yield and should be pretty much simple to remember visually. Okay. So as we mentioned, we have attachment to the muscles. The part that doesn't move is the origin. The part that moves is the insertion part. Now, when you attach muscle to whatever thing is moving, you can have different types of attachments. You can have either direct attachment or you can have indirect attachment. So in a case of direct attachment, you're going to have the epimysium, the outside of the muscle, that is going to fuse or connect directly to the sheet of connective tissue that is surrounding the bone. So that's the periosteum, right? Periosteum is that very tough, dense connective tissue around the bones. This is direct connection. In case it is attached to cartilage, then this is going to be directly attached to the perichondrium. So perichondrium is that membrane that surrounds the cartilage. Now, besides the direct attachment, you can also have indirect attachment, so that in that case, you're going to have some type of ropes or sheets of attachments from your muscle to the place that is attached to. Like, for example, these structures are going to be looking like rope-like tendons, right? So remember, tendons is muscle to bone, right? So you can have tendon attachment, or you can have some other form of a sheet-like attachment that is like a fine sheet that is more or less like a tendon, but is not like a rope, okay? So this is called aponeurosis. I have no idea where they got this name from, right? So this is like really all over the body, but it's not really obvious unless you look for this type of tissue. So here, just making sure that we get the connective tissue that is surrounding the muscle. So on the outside, you do have your epimysium. And then here, you're going to have tendon that is indirect attachment, right, of your muscle to your bone. And here is an example of this aponeurosis. Those are sheets that extend from your muscles to attachments to other tissues. So those are really finer connective tissue fibers. Right, so a bundle of fibers is called fascicle. So that, how many fascicles do you have here? 1, 2, 3, 4, 5, 6, 7, and maybe one that we don't see behind there. So this bundle of the fibers is called the fascicle. Yeah, we look at the endomysium. So this is really the ones that are surrounding each of the fibers individually. And then when you look inside of the fibers, you're going to find myofibrils that are what is composing the fiber, which is the cell. Now, when you think about the cell for the striated muscle, you think about a very, very long cell that has a lot of nuclei, right? So the way that these cells were formed is that during embryogenesis, embryonic development, several cells fused and made this very, very long cell. So the whole length of this cell is really the whole length of the muscle. Okay, so those are very, very long cells. Now, as you get older, what happened to the total number of muscle cells in your body? Does it? I don't think so. You can get atrophied, but I don't think it lowers. What about people who do a lot of weightlifting, those bodybuilders? Do you think they gain muscle fiber or what happens? It gets thicker. That's the idea. Because you're pretty much born with the same number of muscle cells that you're going to have throughout your life. Isn't that weird? If you think about it, tiny baby has the same number of muscle cells than a fully grown adult. That's kind of strange, right? Yeah. The same cells will stay. But you do have cells that surround the main muscle that are stem cells in case of muscle damage. In case you have the damage, like I was describing with the hole on my leg, couldn't pull my leg forward. So after a few days, those stem cells go to the side of the damage and they fix up those original cells just to bring it back to the original function. Yeah. But then a bodybuilder would have the same number of muscle cells as he or she had like when they were born. That's kind of like strange to think about, right? Okay, so as you can imagine, the muscle fibers get larger, but they don't really undergo mitosis. They don't really multiply. Okay, so here is again just putting one slide, the levels of a structure of the overall muscle. So now we're going to start getting into the microscopic arrangement of these muscles and how is it that the muscle is working. Okay, so basically you always have to keep in mind this sliding filament model, because in order for the muscles to contract, you're going to have filaments that are going to slide on each other to bring this structure at a closer, to make it shrink, okay, get into a closer proximity. Now, structures that are important to keep in mind that are only applied to these muscles, in general. First one is the sarcolemma. Okay, so this is a special name that the plasma membrane of the cells get. Okay, so sarcolemma is the muscle plasma membrane. And also the muscle cell is called, in case of voluntary striated muscle, is called muscle fiber. So that's the same thing, interchangeable. Muscle cell or muscle fiber, that's the same thing. Okay. Now, if the sarcolemma, so sarco referring to muscle, right? Sarcolemma is your plasma membrane. What can you guess? It is the cytoplasm. I mean, sarcoplasm. I already told you. Okay, it's the cytoplasm of the muscle. Okay. So now, inside the muscle cells, you're going to find glycosomes. So those are as for storage of glycogen. Okay. So what is glycogen made up of? Glucose, branched chains of glucose, right? So this is for fast resource for energy. Okay. You're also going to find this in myoglobin. So myoglobin is good for storage of oxygen in case you need it right away, and in case you don't have a very if you want to use your oxygen right away instead of waiting for the circulation to bring new oxygen in the form of hemoglobin, so myoglobin is stored also in the muscle for immediate usage. Okay, so the muscle has these weird looking organelles. Okay, so you have myofibrils, you have this sarcoplasmic reticulum that is a version of the endoplasmic reticulum. Okay, and then you have these T tubules that are also coming out of this sarcoplasmic reticulum. But then we're going to look at their structure and their function, like also in detail. So here, these myofibrils, those are very densely packed within the muscle fiber. So here is your muscle fiber, okay? So then your myofibrils are inside this muscle fiber. And then the muscle fiber has a lot of these myofibrils. And as you can notice here, of course, this muscle fiber is striated. You see those stripes, right? And also notice here that you have several nuclei under this membrane that has a special name, right? The sarcoplasma is your special membrane that covers your muscle cell. Okay. So myofibrils are going to make up these fibers inside of the muscle cell. Now, looking at those striations, so you have to know what these bands are and what they represent, okay? So those striations, they are pattern that you see in the muscle once you look at in the microscope and you look at these muscle fibers. You looked at the muscle fibers in the microscope already in the lab? So it's like second nature to you by now, right? Hopefully. Okay, so what you're gonna see, you have A bands, so those are the dark zones. So if you look here, the dark zones are the A bands. And then you have the light zones, those are your I bands. So you have I and light, so those are your I bands. So let's see here. If you look at your A bands, inside of the A bands, you're gonna find age zone, okay? And you're also gonna have the M line. So instead of just talking about what it means here, we're going to also look at better pictures other than just this whole muscle here. Now, when you look at the I bands, and those are the lighter ones, you're going to have, between the I bands, you're going to have this Z disc or this Z line. And it's interesting that when you're looking at a cross-sectional or a flat piece of muscle in a microscope, you sometimes lose perspective that these discs are really a very flat image of what should be a three-dimensional type of structure. Okay, so keep in mind that it's not a line, it's a disc. Okay. Okay, so here looking at what is really important to know about the sarcomere. First of all, what is the definition of a sarcomere? It's a functional unit for contraction, right, of a striated muscle. And the sarcomere is the space that is located between Z discs. So that's your definition of a sarcomere. That's the side defines one sarcomere. So now you're going to look at the Z disc. Where is the Z disc here? It's in the middle of the I band, so the light band, right? So here you find your Z disc. And they have these proteins that if you look at it from a top view, it kind of looks like a zigzag line. I think that's how it got the Z disc nomenclature, right? But then keep in mind that this thing goes across the whole fiber here. So it's really a whole disc that is on the side view here. So Z disc to Z disc, this here is your sarcomere, right? Okay? So remember, the Z disc is also the middle of your I band, and your I band, you're only going to have here your thin filaments. So it's light because the stuff that is in there is thin, okay? So I bands, you have thin filaments. Now, when you look at the A band, you're going to have your thick filaments, but not only the thick filaments. You're also going to have thin filaments that are inside those A bands. Okay. Now you imagine when you have muscle contraction, your thin filaments are going to move this way. Actually, your thick filaments move that way. So this I band is going to disappear when you contract the muscle. Okay. What about the A band? What happens to the A band? Is it going to disappear when you contract the muscle? No. Is it going to get bigger? No. The A band here is the size of your thick filament. It's going to move this way or move that way because this thin filament is going to be superimposed. It's going to be in the middle of it. But the A band is not going to get bigger and it's not going to disappear either. Okay, the light band disappears. So between this and that, Z, that's your sarcomere unit. Now, what is this M line here? Myomycin. See, I went blank. I was thinking of more of T tubules. They're going to be going between these structures. But still, we're going to see more details on that. Okay, so looking here at these myofilaments, okay, so the most important proteins that make up these myofilaments are actin and myosin. Okay, so actin and myosin myofilaments. Which one is a thin one? Actin is the thin one. You gotta keep that in mind. Okay, actin is the thin one. Myosin is the one that has the head with ATP activity, with ATPase activity. So here, when you look at your sarcomere unit between the Z lines, right, you're going to have the thin area. Okay, that's the I that is not showing here. So we can see this here is your I band, right? So in your I band, you're going to have your thin filaments that are actin, two chains that are going around each other. Okay, so this form of actin is called filamentous actin. So keep that in mind. And then when you look here at your myosin filaments, this is the time you see the thick ones, and you see a lot of the heads of the myosin filaments right there. Okay. So another important thing here with your myosin filaments, that they are connected to each other back to back at this M line. Okay. And what is the line that's connecting the light filaments? The Z line or the Z disc, right? So here, looking at more detail at the structure of the myosin, you see the myosin actually has three important sites for the structure. It has, of course, the head, and at the head, you're going to have the ATP binding site, and you're going to have an actin binding site. That's very, very important, right? Because myosin is supposed to bind to the actin in order to push that structure and allow muscle contraction. So actin binding site, ATP binding site, so this is at the head. The flexible hinge region is called the neck, obviously, right? So here you have a neck, though it's not rated there, but this is a neck. And then you have the tail. That's where you have the long fiber, that all the tails here are getting together, right? And make this thick appearance for this fiber. Okay. So important thing to know, you have actin binding site, ATP binding site, flexible neck. Let's just write down the neck here. Okay. And you have the tail. Looking at your actin structure, basically you have two forms of actin. You can have the soluble form, or you can have the filamentous form. So when you look at actin that is in soluble form, those are called G-actin, G for globular protein. So G-actin if it is soluble, if it is not in a filamentous structure. But then, of course, in the muscle, you're going to find it in this filamentous type of structure. So that you call fibrous or filamentous F-actin. So F for filamentous. Okay. Now, this actin is going to have sites or binding sites that are for the attachment of the myosin head. Okay, but you don't want to have contact of myosin to actin unless you are commanding a nerve contraction, a muscle contraction. Okay, so in order for this thing not to contract without us knowing or commanding, so the binding site of the actin is blocked. Okay, so binding site is blocked when the muscle is relaxed or not contracted, and the protein that blocks the active site is tropomyosin. Okay, so tropomyosin here is going to block the binding site of actin, the myosin binding site on the actin protein. Okay, so Tropomyosin is blocking, and you also have an associated protein here that is called troponin. So those are both involved in controlling binding of actin to myosin. Okay, so just here is going into a more microscopic view of the muscle. So here you look at the sarcomere. So looking within the sarcomere, you have the thin and the thick filaments. And now we're going to see more of this interaction between the thin filaments and the thick filaments. Okay. So besides those proteins already mentioned, you also have other associated proteins, but there are a lot of them. So of course, we're just going to keep it simple to the ones that are really more important. Now, there is this protein called dystrophin. Okay, so dystrophin is going to connect the thin filaments to other structures that are outside of this fiber. Okay, so it's going to attach it to extracellular matrix, and that helps to stabilize this membrane, or the sarcolemma, and helps to stabilize the structure of the muscle fibers. Besides dystrophin, you also have another protein that is called nebulin. So this is important to connect your thin filaments to the Z-disc structure. So nebulin would be around here on the connection here for the thin filaments. And you do have other types of proteins that are involved in holding the whole sarcolemma together to stabilize the whole structure of these filaments. But which ones are the ones you have to know? So pay attention, the ones you have to know. Actin, of course, filamentous form, okay? Myosin and all the parts of it. Tropomyosin, troponin, absolutely necessary, and know what they're working, what are their functions, okay? By the way, dystrophin, there are several genetically inherited diseases that have mutations on the gene that code for this protein. And some of them are very severe, and some of them not as bad. But have you heard of this disease called muscular dystrophy? Yeah, so that would be a problem in this dystrophin protein that would attach your light filament to the matrix outside of the cell. Remember, matrix is outside of the cell, right? So just as a side note on dystrophin. Okay, so now looking at this specialized structure and organelles. Okay, so you have here your sarcoplasmic reticulum. So this sarcoplasmic reticulum is really extensive. It's like it forms these tunnels that go all across your sarcomere. Okay, so the sarcoplasmic reticulum is going to hold calcium inside of it. Calcium is going to be very crucial for muscle contraction. Okay? So again, here you have the structures that is good to review here. The sarcolemma. Here you have your T-tubule. Okay, they have direct connection to the membrane. So the inside of this T-tubule here is directly connected to the outside environment of the membrane. And here the light blue, you have your sarcoplasmic reticulum. Okay. Let's look here. The terminal cisterna is really this part that connects to the membrane, to the outside membrane. So this is terminal cisterna. So there is this other protein that is going to keep calcium inside of your sarcoplasmic reticulum, right? Sarcoplasmic reticulum. The name of the protein that sequesters calcium inside of your sarcoplasmic reticulum, thank goodness this has a very simple name, calsequestrin. I mean, it couldn't be more obvious, right? Does that make a good test question? What's the name of the protein that sequesters calcium inside of the sarcoplasmic reticulum? It sounds really complicated, but what's the name of this thing? Calsequestrin. Don't tell me sequester calcium. Sequester calcium is not the name of the protein. Just shift it, calsequestrin. That's the protein. Why is that important? Why is calsequestrin important to sequester calcium inside of the sarcoplasmic reticulum? Yeah, if you don't have calcium sequestered, you're going to have a lot of involuntary muscle contraction. Okay, so calcium has to be in place away from your microfibers unless you want it to get out there, and the way you want to get out there is by a nerve impulse. Voluntary nerve impulse. So here more about the sarcoplasmic reticulum. Okay, so this is going to be a network of your endoplasmic reticulum, basically smooth endoplasmic reticulum, and those are going to have tubules. They're going to be on the transverse here. Okay, most of them are this longitudinal orientation. Okay, and what is more important to know here? Most important thing to know about your sarcoplasmic reticulum is that it's going to work in regulation of calcium levels inside your muscle fiber. Okay, very important for regulating the level of calcium in the cytoplasm. What do you call the cytoplasm of a muscle fiber? Sarcoplasm. That was easy. All right. Now, when you look here, T-tubule. What is this T-tubule? Okay. This is a protrusion or a hole of the membrane. Sarcolemma is a membrane. You see this little hole here? This is called a T-tubule. Okay, so it goes inside and extends the area of your cell membrane to the inside of this fiber. Okay, now this is important because the inside of these tubes here is really continuous with the stuff that is outside of the cell. Okay, so the inside of this tube here is the lumen of the tube, is continuous with whatever is happening on the outside or the extracellular space. That is important for transmission of your nerve impulse. Okay, so this is going to allow your electrical nerve impulse to reach inside of your fiber, of your muscle fiber. Okay. So what is important to know here? Yeah, okay, so here, when you look at this structure here, you have your T-tubule, and then you have this other structure. What is this other structure here part of? This is your sarcoplasmic reticulum. That's the stuff that stores calcium, right? Now look, the T-tubule goes right in the middle, and then you have this sarcoplasmic reticulum on this side, sarcoplasmic reticulum on the other side. That means whatever impulse is going to come through this T-tubule is going to have to be felt by the sarcoplasmic reticulum that is right close to it. So when you look at this structure and you see hole in the membrane going to T-tubule and surrounded by SR on both sides, this little thing here, you call that a triad. Okay, so it's the area that is formed when you have one sarcoplasmic reticulum from one side, from one sarcomere, and another one from the other side, and then the T-tubule in the middle. That's called the triad. Look at the size of this I band. We should have done this right here. Before contraction, this is fully relaxed sarcomere, okay? And then after contraction, look what happens to the I band. And keep in mind what is happening with the A band, okay? So here age and A in comparison to I and the Z band. So now we see I band got smaller, right? Not because the fibers got smaller, no, the fibers do not get smaller, but the overlap increased. So now this band here got smaller. What happened to the A band? Nothing happened to it. It got closer to the other A band on the other side, right? So nothing happened. What about the A band? Nothing. And Z, nothing, right? So your I band visualization here is the part that shifts in case you have contraction of the muscle. Okay. Now, how to get the muscle to contract. This part has a lot of things that are very, very important, not only for the muscle contraction, but also for the neurological function, right, for the nerve function. So first thing you have to know is that the command has to come from the brain, of course, right? So first you have to make a decision in your head that you are going to move a muscle. Yeah, obvious. That decision has to go down your spinal cord and has to contact your motor neuron cord. Now, we didn't see this yet, but here is what is your spinal cord. This is the dorsal side, that's the ventral side. So here, you're going to have a motor neuron by the ventral side that you're going to have the body here of your neuron, and then this, you're going to have an axon. And then here, you're going to have the end of the axon that is going to get together with your muscle, okay? So here, you're going to have your synapse between your axon and your muscle. You call that a neuromuscular junction, okay? You absolutely have to know that. So your impulse is going to come from that motor center in the ventral side of your spinal cord, and is going here. You're going to have an action potential. It's going to travel down the axon and get to your muscle. Okay, so what is going to happen with the overall picture here? Okay. First thing that's going to happen, you're going to have the cells that are going to receive this impulse, that they have to change the polarity of the membrane potential. Okay, so they have to feel the change, feel what's happening, and change the electricity inside of the cell. Okay, so that action potential is going to cross, okay, from your neuron to your muscle, and here what we call the neuromuscular junction. The neurotransmitter or the molecule is going to activate your muscles to contract. That is called acetylcholine. Okay, and we're going to talk more about that. Okay, so what's happening here? At the membranes, you have these ion channels. So these are both at your neural membrane and at the muscle membrane. Okay, so very important. You have those ion channels. So those are going to be very, very important for the transmission of the impulse. So you have two types of ion channels. One type is a chemically gated ion channel, right? And another type is a voltage-gated ion channel. Now, what's the difference between them? If you have a chemically gated ion channel, the way you're going to open this channel is if your chemical, your preferred chemical that has a very fine interaction with the outside part of this channel, if your chemical is there, then the channel is going to open. Is that simple enough? Yeah, right? What's the name of the chemical? Acetylcholine, ACH. So this channel is voltage-gated ion channel. What ion? Sodium, yes. Sodium. Everybody knows that? You have to know that. Sodium channel that is closed without acetylcholine. So acetylcholine is going to get there. Channel is going to open. What is going to happen to sodium? Where is sodium? More concentration of sodium. Yeah, outside of the cell, right? You have more sodium outside of the cell than you have inside of the cell. And why is that? We talk about sodium-potassium pumps. Have you heard of sodium-potassium pumps? Does it ring a bell? Yes, the sodium-potassium pumps is present in basically every type of cell, and it's what is needed to keep the cell alive. If you don't have a sodium-potassium pump, then the cells are going to burst because of osmotic force. Okay, so now once you have more sodium out than in, once you open this sodium channel, with your acetylcholine, then sodium is going to rush in. Okay. Now, besides this chemically gated sodium channel, you also have a voltage gated sodium channel. Okay, so this is really important when you're sending a signal down the axon of a neuron. So those voltage gated sodium channels are very much different than your chemically gated channels. One main difference is that the voltage gated channels has this little ball stuck to it at the intracellular part, and this works as an inactivator of the channel. So the channel is really fast. The channel is going to sense a change in the voltage inside of the cell, like meaning it's not going to be as negative inside of the cell, okay? And then the channel is going to open, going to allow some sodium to go in, and then this ball is going to get stuck to this pore and block the sodium from going in. Okay, so two types of sodium channels, two types of ion channels, okay? So let's see what's going to happen here. So you have your nerve cell, right, that is coming from the spinal cord. Here is the axon of your nerve cell, and then this axon is going to get to the muscle and is going to make contact with the muscle at the neuromuscular junction. Okay? Is also called, the neuromuscular junction is also called motor end plate. Same thing. Oh, and here would be another question for you, right? In order to receive a signal to contract, each skeletal muscle must be served by what? What has to happen for the muscle to contract? All right, how many people think A? Or B? B. Everybody awake? Yes. So it has to be served by a nerve. All right. All right, so let's see what happens in the nerve. This is really cool stuff, right? So at this axon terminal, okay, you are going to have this little bulb thing, okay, the end of the axon, and here is where a lot of this action is going to happen. First, you're going to have these little vesicles. Remember the difference of a little vesicle and a big vacuole? So this is tiny vesicle, okay? Tiny vesicle is packed with this chemical called acetylcholine, okay? Where does the vesicle come from? Where does the vesicle come from? Inside the cell, like where? No, but it's close to the nucleus. Yes, the Golgi. Everybody got that? All right, so this thing, where is the Golgi? Like inside the cell. Yeah, okay, I got it. The Golgi is in the cell body. Where is the cell body of this nerve? This one is actually in the spinal cord, but it's close to the head. It's not close to the muscle. Okay, so this tiny vesicle has to travel from the body in your spinal cord all the way down the axon by anterograde movement, right? Going forward from body to the end. Now, when it gets here, in the cytoplasm over here, okay, you actually have to put acetylcholine inside those vesicles. That's a whole different story. Okay, anyway, let's see. Acetylcholine gets inside those vesicles, right? Now, those vesicles are close to the membrane, close to your synapse. Okay, the synapse is a space between the nerve and the muscle. Okay, so this is your synaptic cleft. Okay, now when this neuron sends a signal that is, yeah. They wait there, but to get there, they had to travel down. Yeah, but then they sit there and they wait to be activated. Yeah, and then you have, like, if you use too many of them, then your cell would have to make more. So then you have this whole process of replenishing those vesicles. And a lot of them are not even filled, they get filled over there. So you always have plenty of vesicles to be filled over there. Except if you put some drug that messes up the whole system and then you die. Which is always the case. I mean, of things messing up the system. We mentioned some cool drugs over here. Okay, so what's happening here? At this junction, right, what's going to happen? Once the nerve travels down your axon, how is it the nerve's going to travel? Action potential, right? Action potential. Change of the inside charge, the inside of the membrane, right? So the nerve gets there, the impulse gets there. That impulse is going to cause some channels that are on the membrane of your axon here, some channels to open. Actually, these guys here, they are calcium channels. They're going to open, and once the calcium gets inside of the cell, it's going to make some proteins in the vesicle to interact with some proteins that are on the membrane right there. Okay? So those are, I don't have the names here, but they're like V snares and T snares. They're like snare proteins. Once they tangle up here because of the presence of calcium, then this vesicle is capable of fusing to the membrane and dumping out their contents in between these two cells here. So contents, acetylcholine, is going to be dumped in the synaptic cleft. Okay? Now, once acetylcholine gets dumped there, it's going to go very quickly to the, what's the name of the channel? Chemically gated sodium channel. This sodium channel has another name to it. It is called nicotinic cholinergic channel. Okay, nicotinic. All right. So you say nicotinic, the stuff that nicotine that you're having in smoking cigarettes. Yeah, that's the name they gave to the channel. Okay. So once it gets there, this channel is going to be open. Sodium is going to rush into the cell. Okay, is going to get in contact with something else. Is going to cause somehow calcium to flood the cell too. So let's see what is happening here. What you have to know, you have the synaptic vesicles that are going to fuse, and then acetylcholine gets dumped. More stuff is happening. Okay? Yeah.
So the vesicles travel down the spinal cord?
They travel. Your body of your neuron is in the spinal cord. Is on the ventral side of the spinal cord. The body of the neuron has the endoplasmic reticulum that goes into the Golgi. Okay, the Golgi is what's going to make these vesicles, and then the vesicles are going to travel all the way down. Yeah. Okay. So let's see here, big picture. Okay. So first, you're going to have, okay, the excitability of your fiber because of this action potential that causes acetylcholine to get in there. Okay. Then that is going to allow calcium to be released from the sarcoplasmic reticulum, unblock the actin, and then it's going to cause your myosin head to get stuck to the actin and get the muscle to contract. Yeah, I know it's a lot. We're going to go into steps. Okay. So let's make sure we understand what is happening here at the neuromuscular junction. Okay, because I know it's a lot, but it makes sense. Actually, it does make sense. So here you have the action potential that got over there to the terminal, right? Action potential got here. That is going to open calcium channels. Calcium channels are going to release acetylcholine right there. Okay, so here is your first stage. First, the action potential gets there. Then you're going to have calcium channels at the nerve axon. At the nerve, it's going to open. Then you're going to have calcium there inside of the cell that is going to cause fusion of the vesicles. And that is going to dump your acetylcholine at the synaptic cleft. Acetylcholine is going to bind to your cholinergic receptors. Cholinergic is for acetylcholine receptor, is another name for it, right? In this case, nicotinic cholinergic receptor. The names keep on increasing, right? Okay. So that is going to cause sodium to rush inside of the cell, okay? And that is going to cause, okay, this is the sarcolemma is the membrane, right? Surgeon to rush inside of the cell, and let's get over there, okay. Oh, so this is not even before the contraction. And what is going to happen, you don't want this channel open for a very long period of time, okay? You want this channel to be open for a very brief period of time because muscle contraction is very fast. So you want to make sure that this channel is closed very soon after it opens, okay? So what's going to happen is that the molecule that is stimulating the channel to remain open, what is the name of the molecule or the chemical? Acetylcholine, right? If you keep acetylcholine on that synaptic cleft, you're going to keep on stimulating the sodium channels. So there is an enzyme in the cleft that is called acetylcholinesterase. And if the enzyme, most of the enzymes end up with A's, right? So this guy is acetylcholinesterase. Guess what's going to do? It's going to break down acetylcholine. Is that a good thing? Yes, because if you don't, it's a big problem, okay? And actually, there are some chemicals that are called organophosphates. You know what they're used for? Have you heard of organophosphates? Those are used for pesticides. Organophosphates block the enzyme acetylcholinesterase, or just cholinesterase for short. Guess what happens to the muscles of the insects when they are flooded with organophosphates? They contract and cholinesterase is inactive because organophosphate breaks down cholinesterase. So sodium channels keep on being open. That means the muscle gets contracted and there is no stop for the contraction. And the insect dies. Guess what happens to us if you're exposed to organophosphates? Well, the same thing. It also, of course, depends on the amount of organophosphate that you're exposed to, right? But basically the same thing can happen. Okay, it's a poison. All right. Keep that in mind, right? Now, this talking a bit more about the acetylcholine, okay? So I'm just going to mention this. When you get your vesicle, and I put a link for the part that I took this video here, okay? This is the Ninja Nerd video, very, very informative. When you get this vesicle that travel all the way from the body, down the axon, and into this axon terminal, so the vesicle is not really packed with acetylcholine. It has to be packed inside there, okay? So what's going to happen is that to get acetylcholine to get concentrated in the vesicle, you have two things going on. You have a proton pump that keeps on putting protons inside the vesicle, and that uses ATP. And then you have an exchange factor, channel, that is going to exchange a proton for an acetylcholine. And this is how acetylcholine gets packed inside of the vesicles. Okay, so here when you got to the end plate, you're going to have this end plate potential. So once the sodium rushes in, the polarization of inside the cell, that before the stimulus inside of the cell was about minus 70 millivolts. So with the sodium rushing in, this thing is going to become more positive. Okay, I'm going to stop here because I'm blowing my time now.